Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 17th February 2021. As we have been talking about the practicals and various experiments that your batch need to perform for this academic session, we are going to talk about all important experiment which is worth for 5 marks, that is paper chromatography experiment. And the main aim of using this paper chromatography experiment is to separate plant pigments using paper chromatography. So the main purpose is to aim is to separate the various pigments, coloring substances that are present in the plants. So aim to distinguish and study the various pigments present in plants through the process of paper chromatography. And theory parts, of course, we know plants carry out the process of photosynthesis during which light energy from the sun is converted into chemical energy. The capturing of light energy is carried out by molecules known as pigments, which are present within the plant cells. Then, what are pigments? Pigments are chemical compounds which are able to reflect only a particular range of wavelengths of visible light. Leaves of plants primarily contain different types of pigments within their tissues. The 4 different types of pigments are listed below in a tabular column along with their colors. So first one is chlorophyll A, and this is dark green. Chlorophyll B is yellowish green. Xanthophyll is yellow, and carotenoids is orange color. So when you perform this paper chromatography experiment, at the end of the experiment, when you observe this paper, all these 4 colors you must be able to see. In order to view and distinguish the primary 4 plant pigments, a simple technique known as chromatography can be used. Now, what is chromatography? It is a technique that is used to distinguish between different molecules. The differentiation is based on these attributes, that is shape, size, charge, mass, absorption and solubility. So varieties of chromatography is there, but one that we are using is the column chromatography. So paper chromatography, column chromatography, partition chromatography and thin layer chromatography. So for class 11th, what we are using is the paper chromatography. In this technique, the interaction between 3 components is involved, that is solid phase, separation of a mixer and a solvent. Now at first the mixer is spotted on the paper and is dried. Second, the solvent is made to flow through the capillary attraction. Third, while solvent moves through the paper, the various components of the mixer differentiate into varied color spots. Fourth, later the paper is allowed to dry and the position of various compounds is viewed. Fifth, the substance which is the most soluble moves further on the paper as compared to other substances that are less soluble. So solubility is the criteria. What are the materials required for this for us to perform this experiment? Chromatography chamber, spinach leaves, mortar and pestle, scissors we need, and acetone, ether acetone solvent we require, acetone is required, capillary tube, small tube we require, tubelet, pencil, spatula, scale, filter paper strips and of course stapler. These are all the materials that we require to perform this. Of course you need watch glass and thread also. And the procedure? In this experiment, spinach leaves are used to separate different pigments. Pick a few fresh and green leaves of spinach and wash it. Cut out small pieces of spinach using scissors, add them to mortar. Accurately measure 5 ml acetone using a measuring cylinder and add it into the mortar. With the help of the mortar and pestle, grind the spinach leaves into a smooth paste. Shift the prepared paste of spinach into a watch glass with the help of spatula. Place a filter paper strip with a tapering notch towards one ending of the strip. That means you have to take the filter paper and cut it one end, okay, with a sharp edge. Place a filter paper strip with a tapering notch. That's what I said. Horizontally, trace a line with a scale and a pencil that is 2 to 3 centimeters apart from the notch tip. Using capillary tube, add 1 drop of extract of pigment in the midsection of the line. Let the drop dry. Repeat the same process of adding drop and allowing it to dry for 4 to 5 times. In fact, when we are performing in the lab, we will do something like 14 to 15 times also. In the chromatographic chamber, pour the ether acetone solvent. Make sure to fold it and staple an end side of the paper. Suspend the strip of the chamber. The loading spot remains about 1 centimeter above the level of the solvent. Let the chamber remain uninterrupted for a while. Maybe we are keeping it for 20 minutes to half an hour. We can notice that solvent passes along the paper, scattering various pigments of the blend to different distances. Once the solvent reaches 3/4 of the strip, carefully take the strip off. Now, go out of the biology lab, allow it to dry in the sun. Observation. The dried paper strip displays 4 different bands, color bands. Discrete pigments can be distinguished with the help of colors. This is observation. Now, what is the conclusion? Conclusion. The keratin pigment is observed at the topmost. Now, the first pigment is keratin at the topmost as orange-yellow band of pigments distinctively. Just below this band, below keratin, yellowish band, okay, appears, which indicates pigment xanthophyll. Third band appearing dark green indicates chlorophyll A pigment. And finally, the yellowish green band is present at the bottom is the chlorophyll B pigment. Now, whenever we are performing any experiment, there has to be some precautions. So here also, for this experiment also, we need to take some precaution. The leaves that are selected should be green, fresh spinach leaves without any disease. From the tip of the notch, the loading spot needs to be 2 to 3 centimeters apart. While suspending the filter paper strips in the chamber, one need to ensure that loading spot needs to be set above 1 centimeter from the level of the solvent. Now, viva questions. What are the viva questions that will be asked in this experiment? What RF value or retention factor? Retention factor or RF value applies to chromatography to make the technique scientific. It is defined as the distance traveled by the compound divided by distance traveled by the solvent. So RF value is equal to distance traveled by the compound divided by distance traveled by the solvent. What is phycobiline? Phycobilines are light capturing bilins found in chloroplast organelles, cyanobacteria and few algae. What is the significance of pigment in photosynthesis? It helps in absorption of energy from light. The free electrons in the pigments present in their chemical structure transfer their energy to other molecules during photosynthesis when they turn into high energy electrons, thereby liberating energy they capture from light. The released energy is then used up by other molecules for the formation of sugars and related nutrients with the use of water and carbon dioxide. Like we always write photosynthesis as CO2 plus H2O. Of course, in reality, they never combine each other. Just for our understanding, we are writing this. Arrow mark, we are writing here sunlight, and then this chlorophyll pigment. So starts with the byproduct, main product, food of the plant. Some water will be released out, that we know in light reaction, Hillman reaction. And then most important, oxygen will be released, which is essential for us. So here this plays a crucial role, chlorophyll. This absorbs sunlight, and you know in theory we have studied PS1, photosystem 1 and photosystem 2. So based on this PS1, PS2, all these photosynthetic reactions take